All right. Okay. So our event this evening is featuring 5 women from Allen Institute that represent 5 different programs at the Institute. So the Allen Institute is an independent, nonprofit bioscience research organization, and their aim is to unlock the mysteries of biology, a small little goal. They tackle large-scale programs, they collect high-quality data, and most importantly, they share their data and resources openly to accelerate discovery. So instead of me telling you about Allen Institute, I'm going to turn it over to our panelists who will speak in this order. And I do have to apologize, Sandra Olak, I have misspelled your name, so my apologies to that. So please take note of your questions during the presentations. We will have an opportunity to answer your questions at the end of the presentation. So each of the presenters will give their little presentation, then they will be invited to come up front, and then we'll start the question and answer period. So at this time, I'd like to introduce Ali Wiener, and she is an education specialist in the Education and Engagement Program. Ali.
All right. Thank you, Retha. Good evening, everyone. My name is Ali Wiener. I use she, her pronouns, and I am an education and engagement specialist at the Allen Institute. I am a proud woman in science, longtime educator, mother to Molly, an avid runner. So tonight you'll hear a little bit about the background, mission, history, and education program at the Allen Institute from me, as well as get to hear from 4 women who work in data, technology, and informatics and engineering across 4 various scientific divisions at the Allen Institute. So first, I'm going to give you a bit of background about the Allen Institute. So we're a biological research nonprofit established 22 years ago by Paul Allen. We're right around the corner on Westlake Avenue. We have 4 scientific divisions that tackle complex, large-scale, fundamental research questions in the fields of neuroscience, cell biology, and immunology, as well as a grant-making division, the Paul G. Allen Frontiers Group, that funds cutting-edge research across the world. And last year, we launched our newest program called the Seattle Hub for Synthetic Biology that builds cutting-edge cellular recording technology in partnership with UW and the Chan Zuckerberg Initiative. So our approach is guided by 3 core principles. They're big science, team science, and open science. And that focus on open science in particular leads to generating data, knowledge, and analysis tools, which we share publicly with scientists and educators across the world. So on the screen there are a few of our open data websites where you can find some of that data. So a little bit about our history. We were founded in 2003 as the Allen Institute for Brain Science with the mission of creating a map of gene expression across the entire mouse brain. We completed that project and we moved on to create a map of the human brain, among many others, and other exciting data projects. And then we decided to expand that model to other fields of biology. So in 2014, we opened the Allen Institute for Cell Science, immunology in late 2018, and the Allen Institute for Neural Dynamics is our newest division in 2021. And so they look more at kind of behavior and decision-making in the brain as compared to brain sciences missions, which currently involves characterizing cell types within the brain. So I mentioned I'm an educator and I'm part of our education program. So one of the reasons we landed here tonight is because Retha, who's a leader in AWIS, is also a local champion of education programs. And so I just want to share a little bit about, there's lots of education programs, both locally and across the US and the world. And so what makes ours kind of special, right? And it is that sort of commitment to open science. So our goal is to take our petabytes of data that we generate from our research and give the public, educators, students tools to analyze and explore it. So within our education and engagement team at the Allen Institute, we use our open data to create curricula for college and high school educators, outreach events like Brain Fest at the Pacific Science Center coming up on March 15th, student field trips and experiences in our building, and a variety of educator professional development programs. And tonight, we're excited to highlight some of our discoveries through the unique lens of women who excel specifically in engineering, technology, and informatics. At the Allen Institute, we are proud to have many affinity groups to help our employees feel included at work. And 20 aims to advance the success of gender minorities in these fields for events exactly like this one here tonight, where our members can network, share ideas, and learn about opportunities for career growth. So you'll be hearing from 4 women across our 4 main institutes in the order that they were founded. So first we'll have Jane from Brain Science come up and share a little bit about her career, her research and advice, followed by Sandy from our Cell Science Division, Kathleen from our Immunology Division, and Mekla from our Neural Dynamics Division. So with that, I'll turn it over to Jane.
Hi everyone, I'm Jane, and just like many of you, I'm super passionate about math, engineering, and computer science. Math helps us understand the world better, whereas engineering and computer science helps us make real-world impact. And where can I make real-world impact? Healthcare. That's why I've been in this exciting field called AI for healthcare for years. And before I jump into that, I want to share a little bit of my scientific journey in case it resonates with you and it really helps you understand why I was drawn to Allen. So it's not moving. Okay, cool. It's starting moving. My scientific journey started from this extremely beautiful island called Taiwan, where I was extremely fascinated by all kinds of scientific experiments and wanted to become a scientist. However, my dreams were challenged when I went through some math camps where I was the only woman, and that was extremely lonely. And so when I was deciding what major I should choose in my undergrad, I decided to go for electrical engineering, partly because I felt like I can still do something with my hands, partly because it's hard to say no to TSMC, the largest semiconductor companies in the world. But then after a year, I finally felt like my imposter syndrome was sufficiently resolved that I started to also double major in math. But that son of a biscuit doesn't really completely went away. So because like I was often the only woman in class again, but luckily I met this amazing woman who was like studying at MIT electrical engineering and computer science. And she told me not to limit it by other people's limited imaginations. So she encouraged me to apply and I did. That's why I traveled to the opposite side of the world and got to yet another beautiful place. Well, I guess beauty is in the eyes of the beholder. To this computer science building at MIT, where I did my PhD. And this is also the place where I found that I have this tremendous power to use my math and computer science and electrical engineering background to make something really cool and make a real impact. So I showed that I can track neurodegenerative disease-affected eye movement features using mobile devices. And the particular idea is that we design an app where we ask people to play this little game. And then like we will use computer vision algorithms to track like interesting features like reaction time or like error rates and see how they're associated with disease progression. And by the way, these people are my PIs. They're not AD patients. But then while I was working on this project, I started to question about, okay, but like I don't consider interventions or treatments. So how would I utilize this kind of information to help doctors design a more personalized treatment per person? That's why I moved again This time, only 30 minutes walking distance away to Harvard, where I did my statistics postdoc. And the particular thing I work on is to design statistical AI algorithms, in particular Bayesian-based reinforcement algorithm, to decide when to send what kind of treatments to help people to like stop smoking and like to reduce drug use by designing. Oh yeah. Anyway, like decide what kind of messages each person should get at each time. And given that I often spend time sitting in front of my laptops thinking about like how to help people become to walk more, it's quite hypocritical in its own way. But anyway, at this point, I feel like I sort of closed this loop that I know how to track disease progression. I know how to use disease progression to design personalized treatment. But then we know that to fully track and understand neurodegenerative diseases, we will need multimodal data. Data like imaging data, pathology data, omics data. And that's why I went to Allen. Or you can say it's because I really love these digit mans. They're my Digimons in some way. So yes, I joined Brain Science last June. So what does brain science at Allen Institute do? Our vision, mission is to be able to understand brain by using a multi-level approach to study cell types. If we are able to understand cell types in the mouse, in primates, in humans, then we can understand evolution, aging, and neurodegenerative diseases, which is where I am in. Well, I'm not in neurodegenerative diseases. I mean, that's what I'm studying. So my mission is to understand the cellular and molecular changes across AD pathology progression by looking at neuropathology data, single-cell RNA sequencing data, and then use machine learning and statistics algorithm to impute some sort of pseudo time, like that is correlated with disease states. And that will also help me and everyone understand what's the relationship between these two modalities. So throughout, I felt like being a woman in science can often feel like this, or I guess not just women in science, but what I found is extremely helpful is communities like this. My grad women in Core 6 community, 20, and AWIS. And this kind of community reminds us that everyone belongs in science and that we need to keep finding ways to include more people in science, especially those who think they don't belong. So now it's a good time for me to introduce you to yet another amazing woman, Cindy.
Hi, everyone. My name is Sandra, but I also go by Sandy, and I am a scientific data engineer at the Allen Institute for Cell Science. So basically, and I'll go into more detail later, but this goal of cell science department is to understand cellular complexity and behavior. There's so many pawns at play within the cell, and our goal is really to understand them on a more molecular basis. And what I do in cell science is I work on maintaining and developing image analysis and cell tracking pipelines. So in more detail, what do we do at Cell Science? In a nutshell, we're really interested in how cell organization, molecular census, cell behavior, and cell environment play along with each other. Again, the goal is to have a transformative impact in cell biology. How do all these things interact? How can we use this information that we get from images and apply them to research and health diagnostics? And so currently we have 4 projects. The first question we're trying to answer is, how do cells respond to environmental stress? The second question is, how do cells form complicated 3D structures? Third question is, how do cells differentiate and move? And then the last question we're trying to answer is, can we program cell behavior? So one important focus that we are looking at is the EMT program. And as a biology refresher, EMT is basically the epithelial to mesenchymal transition. And this is when a cell goes through a transition. In that image you see up there, epithelial cells are usually very like square, right next to each other, and then they undergo changes where they begin to migrate and move, and then they develop polarity, and like how they move and migrate can answer a lot of questions for us. And so what we do is we culture stem cells in vitro and then image them using confocal microscopes. And so in the image below here, we have a 2D colony of EMT. And then one really cool thing we've noticed that in certain conditions, these cells will migrate and change from being just flat spread out on the like matrigel, and then they'll form into these luminoid-based structures. And that's really important for just like figuring out how they like migrate and move and can answer some good questions for us. Another good thing that we give to the public for free, because we are all about open science, is this thing called an Allen cell structure and segmenter. And what that is that it is a Python-based open source toolkit for 3D segmentations of intercellular structures. And you can find it in Napari. And what Napari is it's this program that lets you do image analysis. And so the Allen cell and structure segmenter is a really great tool for researchers and diagnosticians who have these 3D microscopic images, and they want to use segmentation to point out specific organelles or certain features within the cell that they want to focus on. And so this consists of two elements. We have a classic image segmentation workflow that we designed at the Allen Institute, and there's pre-made ones that you can like apply to different types of data. And then one that we're working on that will be fully released later this year is an iterative deep learning segmentation workflow where you put in your data and you can train it and curate it as needed. So now a little bit more about me and my professional journey. I got my Bachelor of Science in Biomedical Engineering from the University of Rochester, and then moved to Seattle, and I worked in a lot of research institutions. I think if you guys have been around for a while, that first logo over there is the Center for Infectious Disease Research. I think they're part of Seattle Children's now, I'm not sure. But my very first job was working as a research technician in a lab that studied malaria. And so when I got this degree in biomedical engineering, I really didn't know what I wanted to do because it's so broad. And so I thought I would dip my feet in different areas. And so this was my first research job, and I really enjoyed it. I grew a lot, but I wanted to move further. And then my next job was that I worked at the Juno Therapeutics, which is now a part of Celgene, I believe. And I worked as a research associate there, where we did work on cancer therapies. And at this point, I was really enjoying myself, but I wanted to do more computational stuff because while I did a lot of wet lab research, I was beginning to become more fascinated with the analysis aspect of it. And I really got exposure at Juno analyzing data that we collected through flow cytometry. So I decided to go back to grad school, and I got a master's degree in bioengineering with a concentration in machine learning from the University of Washington. This is a really cool program. I think it was only a year old when I joined. It was designed for professionals and like people who want to move into industry eventually. And so you could get this degree and then create your own concentration. And I knew I was very interested in machine learning, data science, computational biology. So I just kind of tailored my experience to that. And after I graduated, I got a machine learning internship at the Institute for Systems Biology, where we did work on the gut microbiome. And I used a lot of machine learning and data science techniques to better understand how what we eat affects the gut. And I believe that all of this just kind of led to me gaining the relevant experience that I use every day today at the Allen Institute for Cell Science. So outside of work, I'm a really big fan of the outdoors. I like to say that I love type 2 fun. So I'm also a big runner. I love hiking, diving, anything that's super active. I also love to travel. And so I think it's maybe because I'm always just sitting in front of a computer all day at work, and it's a great way to just blow off some steam. But yeah, I love both extremes of just being like very like educational and like very career driven, but then also just getting out there and having fun. And I think life is all about having a good balance between the two. So here's what I have in terms of advice for women in STEM. These are like very basic points, but I feel like I have like my own insight to it and twists that I would like to tell you guys. So the first thing is obviously networking and connecting with others, which is what you're doing today. It is so helpful to have organizations like this and being able to talk to others. You never know what can arise to these. If you're in school, go to all the open like houses, and like when companies come into town, just go ahead and go to all of those events. Internships is a big one too, especially when you're like in school. Like it's so easy to just kind of forget and like stay like within your program. But internships really help you get that experience that companies look for. So when you're applying for full-time jobs after school or like during school, make sure you also apply to internships and make sure the internships are paid too. Like these days, companies should definitely pay you. Don't accept free internships. They will pay you. And then my last one is apply, apply, apply. And this one's kind of cheeky, but I like to say like have the confidence of a white man, you know? There's this statistic that goes around that like men just kind of like go for it, you know, like how you have all those like lists of things that like qualifications that you need. I feel like as women, we kind of like be like, oh, like I don't fit like I only match like 90% of these. I guess I'm gonna apply for this job. Like even if you have like 50% of the qualifications, just go for it. You'll pick it up along the way. The worst they can say is no. So you never know what can happen. So when I say apply, 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 apply to everything that you see, even if you're only a little bit interested in it, you never know what can lead to it. And with that, that's all I have for you guys. Thank you.
Hi, everyone. My name is Kathleen Abity. I'm a bioinformatics scientist at the Allen Institute for Immunology. So I started my academic training at Rice University, where I did undergrad in chemical engineering, and I was very much started out as a PV equals nRT type of engineer. Especially that Rice was in Houston, a lot of our classwork was in designing distillation columns for refining oil or designing bioreactors, but I did do some undergraduate biomedical research, which I really enjoyed. I also had a chance to intern at Celgene, which was kind of my introduction to the pharmaceutical industry. And that motivated me after college to go work at Genentech, where I was a process engineer for a few years. And worked mostly on process development for monoclonal antibody therapeutics. And this was a really powerful experience for me because it kind of showed me the massive amount of work and team science that it takes to take a promising biological discovery and turn it into a drug that can be taken through clinical trials, manufactured at scale, stored for years, and delivered to patients. And while I was at Genentech, one of the antibodies I worked on was a CD3, CD20 bispecific antibody, which was meant to take T cells and bring them close to malignant B cells to eliminate them. And that got me excited about the kind of expanding field of immunotherapy. And I was very excited about what was going on with CAR T cell engineering, a lot of which was happening in Seattle. So I decided to do my PhD then in bioengineering at the University of Washington, where I joined a lab studying T cell differentiation and generated and analyzed lots of movies like this one here of T cells responding to activation and differentiating. And then towards the end of my PhD, I did about a year Fulbright fellowship at the Weizmann Institute in Israel, where I got to work in a lab developing genomics technologies to apply to cancer immunology. And then I've been here at the Allen Institute for almost a year now. So at the Allen Institute for Immunology, our mission is to advance the fundamental understanding of human immunology through the study of immune health and diseases where excessive or impaired immune responses drive pathology. So we do that through the matrixed coordination of multiple research programs and 3 business units. I work on the computational biology team, and I work on projects in the immune health and aging and tissue immunity programs. One of the really nice, exciting things about working at the Allen Institute and this matrixed organization is that I get to touch and hear about a lot of different projects. But I'll just give you a flavor of one of the things I've been exposed to and am working on here. So is anyone familiar with human immunology research knows, much of what we know about the human immune system comes from blood samples, because those are the samples we can easily take from living humans non-invasively. So one of those studies was recently released by our immune health and aging team, where they profiled blood samples from over 100 donors split between young and old healthy human donors. And we profiled those cells with single-cell RNA sequencing and other genomic and proteomic technologies. And the team then used that data to generate the image I'm showing here at the right, which is a two-dimensional UMAP projection of the transcriptomes of those immune cells measured by single-cell RNA-seq. And you can see that like cell types, so T cells are clustering together in this dimensionality-reduced space, and B cells. And what the team then did is asked, how does aging change our immune cell composition and states? How do these cell states change in response to vaccination or in response to chronic infection? And they had a lot of really exciting findings that are now available, and this powerful data set has been released to the public, which is exciting. But it is well appreciated that though this is very powerful, where these immune cells are actually executing their critical functions for our health is not in the blood, but in tissues, as wisely stated by one of our SAC members, Dr. Donna Farber. So just as an example of that, in lymph nodes, we have B cells residing in germinal centers undergoing this highly spatially controlled affinity maturation to produce functional antibodies to fight disease. In barrier tissues, such as lung and intestine, we have immune cells that are specifically adapted to those tissues and provide the first line of defense against invading pathogens. So given that importance, the Allen Institute for Immunology is putting a lot of effort and resources into using spatial transcriptomics to study a variety of human tissues. And what I'm showing here is one of those data sets for a human tonsil section. And from this kind of faraway view, it might just look like a standard immunohistochemistry microscopy image. But if you zoom in, you kind of appreciate the richness of this data set, where now we've segmented each individual cell outlined in white. And then each tiny little dot in the image is a single transcript that's been detected in these cells for one of 471 genes that we chose to measure in this experiment. And unlike conventional single-cell RNA-seq, we've detected these genes, but also preserved the spatial location of the cells. So we can then ask which cells are talking to which other cells and how spatial organization is regulated. So one of the things that is exciting to me to come to work to every day is I get to creatively think about how we extract quantitative information from these data to address our hypotheses. I'm going to skip these for time. So I'll end. I just wanted to kind of introduce myself in a more personal manner. Like many here in Seattle, which I think Sandra touched on, I love the outdoors and I love doing intense activities out in the outdoors. And this is something that I've actually kind of integrated into my career for a very long time. So this image is from about 10 years ago. I'm the person hanging on at the very tail end of this cycling pace line. And this was a cycling group that I had at Genentech that was an inspirational combination of scientists, postdocs, even the head of business partnering, who I never, of course, would have had FaceTime with, except that we had this nice cycling connection. And this group, I would say, was actually formative in my kind of decision to then go on to grad school and pursue a higher degree. And then when I was in my PhD, I got very into competitive bike racing, which was I think a really nice outlet for my focus, especially when research was not going well. And this was actually something that my lab was super supportive of. And when I graduated, they surprised me with a lab group bike ride, which was very special. And then when I was in Israel for my research fellowship there, I was in this entirely new place with new colleagues, and I found cycling to be this grounding sense of self that I had and I could share with other people. And so that image is a ride I did with my PI there in front and lab mates from that group. So, and then lastly, more recently here at the in the past couple of years, and since I've been here at the Allen Institute, a former mentor of mine from my internship 10 years ago at Celgene, together with her and also my partner who also works at the Allen Institute, we've put together this backpacking women's leadership development retreat that we do every year in the Sierra Nevada wilderness, where we kind of take on outdoor challenges and also discuss leadership development. And I just wanted to end with a quote that is from one of the articles we always read in that leadership development retreat. Think of your life as a house. Can you knock down the walls between the rooms and be the same person in each of them? And so this is something that I think has served me well in my career, kind of integrating the passions that you think of outside of the work with work. And I also think that it's something that I'm really grateful that Allen Institute really supports. I think we're, explicitly encouraged to bring our full unique selves to work and have that be part of what we contribute to work, which is super special. So with that, I'll end and introduce our next panelist. Thank you.
Okay, cool. Hi, my name is Mekla Kapoor. I'm a software engineer in the Neural Dynamics division. Our mission is to understand behavior and decision-making from the individual neuronal level to the neuron network and circuits. So, and the way that I contribute to this mission, and honestly, the way my team contributes to this mission, is by developing, maintaining, and deploying software to accelerate the collection, processing, and analysis of large-scale neurophysiology data. And that is my team's backs, if you ever recognize us. So the way that we organize our science in the Neural Dynamics division is by platforms and projects. You can think of our platforms as the ways in which we are standardizing the collection and processing of data, while our projects are the questions that we are answering with said data. And as you can see, it's not a one-to-one relationship. A single data platform is utilized by multiple projects, and multiple projects are utilizing multiple data platforms. And the reason this is because while these platforms are standardized, there are multiple choices made that affect our scientific question. I'll use like the NeuroPixels electrophysiology as an example. That platform is collecting neural recordings across the brain, but where those neural recordings are coming from is important for that scientific question. So since there are different brain regions, that information is really important. And the way we think about this is it's called metadata, which is the data about the data. So the brain region is kind of one part of that metadata that lets us explain and utilize the data that we've actually collected. And if you want to learn more about our platforms and projects, you can check out our website. But the reason that I wanted to bring this up is because as a software engineer in data infrastructure, we are primarily focused on the metadata infrastructure to support those data platforms. So how can we build something around that metadata to make our data actually usable and reproducible by people within the Institute and outside of the Institute, and really support those projects. And the data infrastructure supports the life cycle of data, and it does so across a wide variety. And we're going to go through that life cycle by following surgery metadata particularly. So as I mentioned, data infrastructure supports every stage of the data life cycle. Our metadata is stored across multiple databases, file systems, and probably the hardest thing to get, the information that's stored in our scientists' heads. And what our job is as software engineers in data infrastructure, so you can imagine me in that question mark, is to standardize, compress, and transfer all of that information from all these private sources and upload it to the cloud. And I really want to highlight the reason we're uploading it to the cloud is to support open science and team science. So if our data is going from these private assets to a public platform, we can then work together on data analysis and ask new questions. And those questions can really be like we can continue asking questions by utilizing the same data sets. So the way that our searching metadata lives is it lives across about 5 different databases at the Institute, and we ingest that metadata through one of our software tools that I've worked on, the metadata service. And basically, what this does is it pulls all of this information from the databases and consolidates it into, it consolidates it into a JSON document that is our standardized format. That JSON document then gets uploaded to the cloud where it's hosted in an Amazon S3 bucket that is open. So again, this allows us to really share our data with people within the Institute and outside of the Institute. So whenever we go to like UW or University of Arizona or any other institutions for workshops, we come prepared with our data ready to be played with and ready for new questions to be answered. So it's really exciting work. And with that, like my key takeaway from my career is that science is super interdisciplinary. So as I mentioned before, I have a bachelor's degree in computational neuroscience, but when I graduated with that degree, I kind of was faced with the option of, do I pursue research? Like, do I go to graduate school, pursue a research topic, a PhD maybe, or do I try to dip my foot into the industry? And I decided to go the latter when I found the Allen Institute, and I really appreciated it because it's taught me that I'm able to support research and multiple scientific questions through working on data infrastructure, and that's really rewarding. So yeah, that is my main takeaway. And I had a couple other images about myself, but they have not shown up. But things about about me, I am not a blank slate. I do like art. I am an artist outside of work and have gotten to pursue that artwork once a year for our Halloween costumes, hence the psych comp. Thanks. Boxes. I also am a cat mom, and yeah. Thank you. And